All right, thank you for joining us this morning, everyone. My name is Bob Bruner. I am the Exotic Forest Pest Educator with Purdue University Department of Entomology. This is our third session of the Emerald Ash Borer University. This is a program that is done in partnership with Purdue University, Michigan State, and Ohio State universities. And today we have Bina Thapa here from the Department of Forestry and Natural Resources at Purdue here to talk to us about Emerald Ash Borer. And then Bina, do you want to go ahead and take it away?
Thank you, Rob, for your introduction. Hello, everyone. I'm Bina Thapa from Purdue, like he just mentioned it. I'm currently postdoc with Dr. Brody Hardiman and Dr. Sang Fei. And then my research interests mostly on application of remote sensing in forest management. But today I'm going to talk about the emerald ash borer host mapping in forest. So basically, we know that like EAB has caused a lot of damage in the forest. Here you can see a lot of like dead tree of the ash tree near to the Canadian border. So it costs more than $10 billion for treatment, removal, and planting in the U.S. alone. That has been the research done by the Kovacs et al. 2010. Not only in the natural forest, it also has affecting the trees that are growing in the urban areas. Here you are looking at the ash tree before the outbreak and after the outbreak. That's basically killing all the ash trees from the right side to the left side to the right side. So from the study from 2010 from Kovacs et al., you can see a lot of like urban trees in the residential and non-residential area has also been affected, and it has been causing a lot of like economic cost. Not only the economic cost, it also caused the damage to the ecosystem as well. It also affect the aesthetic value and also having the ecological like carbon emissions. So what's the emerald ash borer? Like basically it's an invasive species, has been introduced from the Asian to the US state. Like initially it was detected in 2002, Michigan. This invasive species only affect only host tree species. What does it mean? Is only killing the trees that has been it lives on, such as white ash, black ash, green ash, and blue ash. If you have an ash species, then only this species only affect on this host species. So this is just the maps just showing the current distribution of the EAB. You can see a lot of areas that has been infested, detected since 2002 to 2022, and then current distribution along the Minnesota regions in here in purple color. So in Indiana, it has been detected since 2002. So that means the forest ash tree species within the natural and urban areas both has been affected by the EAB. Before going into the detail, I would just like briefly go with the life cycle of the EAB. We know that like EAB basically emerges in early May. They has the lay eggs between the May and September, and those eggs convert into the larvae stage and then go to the pupae, but most of them are like overwintering and then emerge into the either next year, depending on the temperature, or like 1 or 2 years of the delay. Most of these EAB travel only small half miles per year. They cannot travel a lot like long distances like the other species. Like I had, if you know about this Spitsbottom, they can travel like 100 kilometers based on the wind direction, but this only can travel like half miles per year. It has been spreading slowly from the eastern to the western regions, definitely. So here you are looking at the EAB typical damage. Like basically when I say like those like adult EAB emerge from the tree, they make like D-shaped exit hole like in here within the red circle. And then the other side of the figure, you are seeing the typical damage of the EAB. It's like most of the time they feed on the leaf, and also they made a gallery within the cambium. So once the larvae make a cambion within the damage the cambion layer of the tree, it basically stop the nutrients flow, and then you can see the crown damage at the beginning, and then slowly after a couple of years you can see the whole tree, a big whole tree is like dead. But in some cases, the ash tree can show the symptoms by developing the epicormic branches, and it's kind of like it's grow back again, like in this picture. So what are the factors basically affecting the damage and extent of the EAB outbreak in US? It basically depends on the host availability, natural enemy, and then weather. So to manage the damage, to minimize the damage of the EAB, we need to know the spatial distribution of the ash in US. So once we know the distribution of the ash, then we can regularly monitor those forests and then identify if the trees has been infested or not, and then what kind of devise the different management strategies. So in this talk, basically, I'm going to talk about the two projects that I did. One is the during my PhD, in which I used the Landsat data and EFI data to map the a lot of different species, but one of them is the ash species. The other one is the one with the planetoscope data and then individual tree data, which I did last year during my postdoc, and then mapping the species distribution in the Cook County, but one of them is the ash species as well. So first, I will just begin with the natural forest, mapping in the natural forest. Like I mentioned, I use basically Landsat data and EFI data. It has already been published. So Landsat data, if you look at the Landsat data, is like it's not very high resolution image. It's like 30-meter spatial resolution, but it is enough for the natural forested area. If you look at the Landsat spectral, then you can see in here the chlorophyll absorption regions and water absorption regions, and then with these different kind of bands, I have like calculated a lot of vegetation indices. Vegetation indices, sometimes it's just the band combination of one or two, or like sometimes like multiple band combinations. You can find a lot of reference how to calculate the vegetation indices in remote sensing data as well. So I calculated a lot of these vegetation indices and then all the bands available for the Landsat data, and kind of link with the ground data, this EFI data here. So this is the general plot overview of the plot layout in the EFI data. I have used the EFI data from 1990s and 1997, which is periodic. You can see all the red circles are the periodic EFI plot design. And also I use the annual EFI data, which is in the blue color. And then you can see the like Landsat pixel overimposed over those FIA plots. So in this study, basically, I also try to figure out which FIA plot is more aligned with the Landsat data to calibrate the model. So these species that I had used were like ash, aspen, maple, pine, and then the major focus was the defining the Spruce-bottom horse species. So this is the study area in which I had like 6 Landsat footprint, like from row 26, 27, and part 26 to 28. So I use all the images available that's like cloud free, and then capture the phenology of the species within these regions. So here you are basically seeing the phenology of these species. Here, the phenology of the black species. In early July, they are like all green, and in early September, they are like they leaf off. So the phenology of the ash species in Minnesota is like live on later than the other species, but live out earlier than the other like maple or like aspen species. So that's the unique phenology that I intend to capture with the Landsat image data and then link with the FI data to develop the model. So the model I used was the PLS regression, in which basically used the all the variables. Variables means Landsat band and then vegetation indices. So as I mentioned before, from the images that are available 1980s to 1990, and then for 1985, and then for 2000 to 2008 images for 2005 with the annual FIA data. So use those variables, did the forward selection method, basically picking the which variables is very important for the model calibration. So each time it drop the one variable, calculate the model RMC, R square, and then select the optimal set of the variables for the final model development. Here, once we know like the variables are picked, but we want to know which variables are very important for particular species. In the left figure here, in my left figure here, you can see the PLS loading factor. Basically, it says that if the value is high, it doesn't matter either negative or positive. If the value is high, then those bands or images are particularly important for a species identification. The right figures basically summarize the all the importance variable picked for selected based on the models, and then I distributed according to according to the basal area and then species wise. So here you can see here that as a species, a lot of like MSI bands are in vegetation index was very important, and not only in the as a species, across all these species, those vegetation is very important to predict the species within the regions. So this is a little bit more detail of the score loadings from the previous slide. So you can see here, for the asset species, I have used the star sign. Once their predictor variables is like greater than score is 0.8, then all the star sign was used in an asset species. Basically MSI index, NBR index, and then tassel cap, and then band one indices. So yes in here basically denotes the image from the September date. Similarly, you can see other species has the Mars. For the first spruce, it is from the Mars image, and black spruce also request the Mars imagery as well. So here in the x-axis is basically all the species, in y-axis is the predictor variables that are very important and in identification of the species. This one is the big table. I don't want to go all over the all the details with that part, but all the like bold here indicates the R square importance. Indicates the good model across the part 26, 27, and 28. So I hope I hope you remember, like I have like study site that includes different paths across the study area. So is across all this Landsat path. Then I have like for birds model example 52 has the model accuracy of R square 0.52, and it comes from the path 26 only. In case of the ass, we have like path 27 has the good model, this has 0.64 R square. And then in the bracket is the sample size, and then the other one is the cross validation of the model, and then test RMC is like very small in here. This is for 1985 models that I have developed. Similarly, in the next here, you can see the model calibration results for the 2005. Here, instead of the 27, I have the better model from the part 28 that has like 0.59 model R square. And it is a little bit small, it is slightly small than the 1985 model. So like I mentioned earlier, all the bold numbers indicate the models that is best across the path. From here, you can see all the models that has the highest classification, highest accuracy is Blacker spruce, spruce, and conifers as a whole. So in the bottom here, BLF represents the broadleaf species, CON represents the conifer, and SPW host is the combination of the spruce and forest species. But for today's specific topic, you can see the R square, model R square for the Asa species varies across the Landsat path. In here, you're basically looking at the relative basal area of the Asa species within the study area. You can see a lot of dominant of the Asa species in the southern part of the Minnesota and around this close to the wilderness area. Similarly, in 2005, the presence of the Asa species shifted from this particular regions, from the top left corner, and then it has shifted towards this part. The next is next regions, but the dominance of the Asa species in southern part of the Minnesota remains same. So which means the dominance of the Asa species has been increasing, but also changing from 1985 to 2005, and a lot of EAB outbreaks was happening in these particular regions. So from this analysis, what we found is like not only the availability of the image, which is capturing the phenology of the species, but also the relative abundance, and then where is the tree species, either they are like overstory or understory. These factors affect the overall model accuracy. In here, you can see a lot of you can see the abundance of the Asa species across the path from 1985 to 2005, and then you can see the number of the Asa species increasing in 2005 as well from the field data. Now, like going back to the urban forest, in case of the urban forest, we know like our Landsat pixels cannot capture the individual tree species level. So for this part, I basically use the planetoscope data, which is which has higher spatial resolutions. For this one, I use the 3.2. Planetoscope has centimeters from, I think, 30 centimeter to some 3.2 meter spatial resolutions, but the one that is freely available is like 3.2 spatial resolutions. And then planetoscope data basically collects the everyday, but because of the cloud within the regions, is only the data I have is almost a week or 2 weeks apart, depending on the seasons as well. So in this study, I used the 2 regions within the Illinois. One is the northern part of the Cook County, as you can see in the red, within the red rectangle. The another study area is the DuPage County, which is denoted by the blue rectangle. So all the individual tree locations are denoted by the gray circles in here. These three data were collected by the Morton Arboretum and the Arboretum from 2013 to 2017. Planetoscope data I have used in this analysis is from 2021. In this study, I particularly focused on the from one images from the single year so that we do not have the confounding effect from the phenology shift in the phenology shift from one year to next year, which kind of like might create confusion in the model calibration. This is generally the overview of the workflow for species identification that I did in the urban area. Basically, here you are seeing is like I have like images from March to all the way to November, and all this planetoscope image has only 4 bands, and use those bands to calculate the vegetation indices. So I could have a lot of like image predictor variable from the single image, and use the field data at this species and genus level, and combine those to select the combine those and then select the important features based on the random forest. Since I have like images from almost a week apart from a week apart, and then I calculate all the bands. I use all the bands and vegetation indices, so I would have like more than 200 predictor variables from this from these images. So rather than using all 200, I use the random forest to select the important features. I use those important features to classify these species and genus to separate model here, and then also compare the random forest support vector machine and neural network, and then compare their model performance across the species and genus level. Once I have the best model, I use the best model to predict the map. Besides that, I also use the test the seasonality effect on the model calibration here. So I use the own images from the spring seasons, summer seasons, autumn seasons. I also combine with the spring, summer, or like autumn, spring, or like combine all the images, and then evaluate its performance for species and genus classification. So like I mentioned, I use the random forest feature selection. So for each species and genus here, I have like all the features selected, 50 important features, and then some of them are common and some of them are not common across the species and genus. And then in x-axis, you can see the image, date of the image, and then vegetation indices. And then it's colored by the spring season in the green, summer in a blue, and then autumn seasons for like red colors. So from here you can see a lot of images from the from autumn seasons were selected. And then if you look at the y-axis, that's a species and genus. So and then all the variables that are common in both are indicates that the model, those indices from the births from particular date, and then date is very important to identify the species and genus. So the range of the color in here also indicates the relative importance of the image variables in species identifications. For instance, here you can see the main 12 vegetation index 4. That's basically I calculated by using the band 4 and band 1. It's very important for both species and genus model calibration. So a little bit looking into detail of the vegetation index, how does looking at the vegetation index across the different seasons. In the x-axis is the image date I had about image date I used from the planetoscope. In a y-axis is the autumn index. That's the vegetation index I vegetation index calculated based on the red and blue band of the planetoscope data. And their equation is and also indicated in a y-axis. All the box plot represents the different species. In here, you can see on 21 species. So each color is if you are looking at the red colors, those are the Acer species. And in Fraxinus species, which is ass species, here I have is just the Fraxinus americana and Fraxinus pennsylvanica, like green ass and white ass in the pink color, and then pink color in here. So you can see there is a change in the remote sensing like phenology indicator throughout the time for each individual species. But in here, if you just focus on the autumn seasons, then you can clearly see how as a species their value is clearly important, like their signature is like very different than the other species. And from October 1st, their indices value is almost similar to the other species, but it's kind of like increasing is the color, leaf color changes of the acid species. And then it peaked on October 23, and then it slowly leaf off after the during the November, and towards at the end of the November, this value almost similar to the other species. This is just the example of the capturing the phenology of the species by using the remote sensing data. And then autumn index may work for the all species, or also may work or may not work for other species. So that part of the analysis that we still need to do it. But for us species, especially in Cook County, is definitely capturing the unique phenology, and then its spectral is very unique, and which is very helpful to identify these species. This is just a classification results. From here, you can see overall classification results from the for both species and genus. The blue color represents the species model, and then orange color represents the genus. In x-axis, in a figure A, you can see the different model comparisons: random forest, neural network, and support vector machine. In a y-axis is the classification accuracy. From figure A, you can see support vector machine is performing well than the other species. Then other models, random forest and neural network. But if we are looking between the species and genus models, then the genus model is performing better than the species model. In a figure B, you can see the image. If we combine images from all seasons, like in here, in this case, 3 seasons, then classification accuracy is higher for genus and species as well. In the x-axis here, you can see the classification accuracy. In a y-axis is the all seasons, individual seasons, and combination of seasons. From here, you can also see the images from the autumn seasons and spring seasons are very important, or like autumn seasons or summer image also very important. That's basically indicating we need autumn images to capture the phenology. Or we can say the species that I have used in this model is has the unique autumn phenology, and then that's why we need the autumn images to classify these species. Here you are looking at the classification results. For species in the left figure, and then genus in the right figures. In the diagonal, you can see the diagonal represents the classification accuracy, and off diagonal is the misclassification accuracy, or like you can say the confusion matrix. Here you can see the classification accuracy of the both Fraxinus pennsylvanica, which is green ash, and white ash like americana. Their classification accuracy is very high, more than 0.8. In both cases, at the individual species level and also a genus level. In this map, you are looking at the as the species distribution that I created for the Cook County based on the support vector machine. Here you can see a lot of highlighted regions are within the natural forest, and then some of the area are within the suburban region of the Cook County. And black color is represents the other species or like non-forested area. Just looking at into the little bit detail here, you can see a lot of dominance of the other species in suburban regions in the one here in top left corner, and then similarly in others other close to the urban areas like urban areas in close to the Chicago regions area in here in the bottom left figure. In the top right figures is the within the natural forest. From this figure, you can see there are a lot of asterisk species distribution within the Cook County, and we definitely need to be more concerned about the susceptibility of the EAB outbreak. Similarly, I also created a map for the DuPage County. Here you can see a lot of like dominance of the as species in particular regions in here, and then here as well. So besides this one, individual asset species, I have created the map for all the, since I used the model by using all these species. So I created the map for these species across the regions, across the Cook County. Here for here, and this one at the genus level. I think I am a little bit ahead of the time, but I would like to thank you all. I would like to acknowledge all this funding agency that has supported me through for this study, and also like to all the co-authors that I haven't listed here, but Lindsay Dennis is the one who has been helping me in the planetoscope data, and then my previous collaborators from the Iowa State. And then I will take questions. Thank you.
All right, thank you, Bina. That was really interesting. As a drone pilot myself, I'm always curious to see where research like this is going to intersect with UAS technology, as I've done a lot of drone flights over areas just like that, exploring ash tree damage and what it can look like. Do we have any questions for Bina? All right, I am not seeing anything. So, Bina, thank you very much. That was really, really interesting. And I'm looking forward to where else your research is going. I know a lot of the work that you do with the Songland Fay Lab has some interesting results coming out of it. And I'm definitely looking forward to seeing what else is produced there. Other than that, I'm just going to send out the recording as soon as it's done, and we can get out a little earlier today, so that's just fine. All right. Thank you, Bina, and thank you everyone else for coming today. You guys have a great week.
Thank you. Thank you for inviting me. Thank you, Bob.